Okay, so in class, what we were talking about is kind of the way in which metabolism gets regulated. Okay, so these are the notes that kind of go along with that. And this is a little bit of more of an elaboration. And certainly, if you have questions, we're going to go over it again in class. So basically, the main idea is that we have a lot of metabolic reactions inside of our body, obviously, that need to be regulated because it doesn't make any sense to run these things on high when we don't need any of the things that they're making. We would just be wasting energy and resources and things like that. So what basically happens is that our metabolisms get regulated in response to the cell's needs. Okay. So before we get started, what I want you guys to do is just take a second and look at this from a TOK's perspective. Okay. And it says many metabolic pathways have been described following a series of carefully controlled repeated experiments. So to what degree can looking at the component parts give us the knowledge of the whole? So think of this kind of as that discussion that we had earlier in class, way back probably it was in, I don't know, maybe September, where we were discussing kind of looking at it from a reductionist standpoint, kind of taking it from the outside and kind of looking in, or the other way to do it is kind of look at the little bits and pieces and kind of work our way out so that we can see how this whole component is kind of a function of all of the smaller parts of it. So just go ahead and take a second, write that down, and be prepared to share out in class. Okay? So as far as metabolism goes, we said, of course, this is all the chemical reactions that are going to take place in our body. And it's the way that the cell is going to kind of manage its resources, manage its energy, manage the materials that it has available to it, as well as the waste breakdown and so forth, and do so in a way that's kind of conducive to its own well-being, so it's not really wasting resources and energy on things that it doesn't need. All right. We also discussed the idea of enzymes kind of working to lower the activation energy. So for instance, if you look at this black line right here, this represents the enzyme and the amount of energy that would be required in order to run this reaction in the absence of an enzyme. And you can see, of course, this red line here, this is the same reaction in the presence of an enzyme. And you can see how we're lowering the amount of energy that's actually needed. So what this is doing is lowering the activation energy. It's making the reaction kind of more conducive to running, say, in this instance, like maybe perhaps at like a biological temperature or something like that. So the key point there, of course, is just that it's lowering the activation energy. And you can see that graphically with the amount of free energy that's needed in order to make this reaction go. Okay. We also discussed some of the things that are involved, or how this, rather the enzymes get involved in making these reactions go. So we say that sometimes this active site, this spot on the enzyme where the chemical reaction occurs, the active site can act as a mediator, brings kind of things together, allows them to react more favorably than they would if they were, there was no enzyme available in the solution and we would just rely on these chance collisions. So the enzyme is going to help to speed this up. The other thing we said that the enzymes can do is they can stretch the substrate molecules' bonds in a way that makes it more energetically favorable for them to react, kind of moving them more towards their transition state than they otherwise would just in the absence, of course, of an enzyme. We said that the enzyme can make the microenvironment more favorable, in that what it's going to do is it's going to maybe kind of exclude some of the things that might be present in that reaction mixture and keep them from reacting. And then we also said that it might briefly participate in a covalent sense in the chemical reaction. And of course, keeping in mind that the enzymes themselves aren't going to get used in the chemical reaction. A single enzyme can catalyze many hundreds, thousands, millions of reactions in a very short period of time because they're not being consumed. They're just repeating their job over and over again very, very rapidly. But they can perhaps, the fourth way here is just participate in the chemical reaction, making it more favorable, and then quickly taking on their native shape and getting back to being able to perform another chemical reaction. Okay, so enzymatic reactions, of course, involve molecular motion. We talked about that. We discussed the reasons why, for instance, like heating up a chemical reaction can make it go quicker to a point. Of course, if we denature that enzyme by adding too much heat, then the reaction isn't going to work properly. But we said that there are, certain optimums, of course, and if we're operating within the functional range of that particular enzyme, by heating it up, generally speaking, we can speed up that chemical reaction because we're going to force these molecules to not only collide with one another more rapidly, but also collide with those enzymes and kind of speed up this reaction as a result of that. Okay, So we talked about the temperature, we talked about the salt concentration, the pH, all these types of things have an influence on the enzyme's function. Because if you have too much of any of them or too little of any of them, depending on the type of enzyme that we're talking about and the normal place where it's found, anytime we get out of this optimum, we're going to affect the secondary and tertiary structures of the enzymes, and we're going to affect, of course, that reaction rate. So cofactors, what they do is they help affect the enzyme's function, and what they are basically are the minerals that we're getting, say from our diet. So these micronutrients, things we talked about earlier in class, metal ions, for instance, like selenium or something, that can be present in very small amounts, but they're cofactors that are going to help this enzyme do its job more efficiently. And then we talked about coenzymes, which also help with enzymes, and these are organic substances. They contain carbon and hydrogen, often nitrogen, things like that. And these are organic molecules that we refer to often as vitamins. So when we get our vitamins by either taking a multivitamin, we can get our cofactors from those too, but we get our coenzymes from either taking a multivitamin or eating a kind of a healthy, balanced diet. And generally speaking, in developed countries where food is plentiful, people often get all of the types of things that they need just from their particular diet. All right, so things that affect the enzyme's function, of course, we said that if you increase the temperature up to a point, you can increase that rate of reaction. And if we look at the rate of reaction right here, what we can see is that it's increasing, but as it gets to this point right here where it's starting to kind of level out, this is where all of the active sites now on the enzymes are becoming saturated. And the only way that we can really speed up this chemical reaction now is to add more enzyme to it, to make more active sites available. And then that would kind of speed up that reaction. And after a point, now that we're saturated, reaction is going really fast, at a point now it's going to start to slow down, because what we're going to do is we're going to start to use up all of the substrate molecules that are present in that reaction mixture. Okay, so that saturation point is something we discussed, something that you want to make sure that you understand how to look at the graphs and kind of understand what's going on, reading the axes and knowing what that's telling us. The next thing that we talked about were inhibitors, and we said that what inhibitors basically do is they inhibit things. In this case, they're substances that can either slow down or stop the enzymatic activity altogether. And there was two main types. We talked about competitive inhibition, non-competitive inhibition. And what the competitive inhibitors are going to do is they're going to compete with that active site. These are molecules that are going to compete directly with the active site. They're going to compete with that substrate molecule, and they're going to slow that reaction rate down. It'll eventually, kind of get to the same point at saturation. It's going to take longer to get there, though, when that competitive inhibitor is there. These non-competitive inhibitors, rather, now these are going to bind to a spot other than that active site. They're not competing with the active site at all, but they're changing the shape of the active site just by binding to it. And that's going to slow that reaction rate down. It's going to bring the saturation rate down as well. So if we look here kind of at this graphic, what you can see is with the normal binding here, what we've got is the substrate molecule is going to bind to this active site, and it's going to get catalyzed into the products. Whereas if now here we have this competitive inhibitor, these two things are going to compete directly with one another for access to that active site. Okay, so that's going to slow the reaction rate down. And then down here with this non-competitive inhibition, you can see this non-competitive inhibitor is going to bind to a spot on the enzyme molecule somewhere, and it's going to change the shape of the active site. So it's going to bring the saturation level down. It's going to slow the reaction rate down. It's going to bring the saturation level down as well. So we looked at a graph that looked like this, and this pink line is kind of the normal way in which that enzyme is going to work. And then when we add that competitive inhibitor, you can see here we're slowing this down, but we're ultimately getting to that same saturation point, because as the competitive inhibitor and the non-competitive inhibitor are kind of interacting with that active site, there's always some active site available, so we can eventually get to that saturation point, or we can elevate that rate of reaction up to the point of saturation. Whereas with this non-competitive inhibition, what we're doing is we're changing the shape of the active site. So that active site is no longer available, even though the enzyme is. The active site is no longer available, so we're decreasing this saturation point down because of that. Okay? So as far as now regulating enzymatic activity, there's different ways that we can do that. And one of the common ways is just to change the shape, and that's kind of some allosteric regulation. All right, so it's kind of like a form of non-competitive inhibition in that it's changing the active site, and we'll see kind of how it works here in just a second. But what we're doing is we're getting a regulatory molecule that's going to bind reversibly to the enzyme, and it's going to change its shape when it's bound. So in some cases, that binding can kind of speed up, change the active site to a shape that actually works. And in other cases, it can bind to and change the active site in a way that makes the enzyme not work. And the key point here is with allosteric binding is that we're going to get a binding and it's going to cause a change in the shape of the enzyme, either making it more active or less active. So with allosteric inhibition, this binding molecule changes the shape of the active site, it inhibits the reaction, and it slows or stops the enzyme's function. Okay, and oftentimes the allosterically controlled enzymes have multiple subunits to it. Not always, but oftentimes it does. And then what we can also have is an allosteric type of regulation where we get binding to a particular subunit or binding somewhere on the enzyme, and that's going to speed up the active site. Okay, this is the allosteric activation. So this is when that molecule, again, is going to reversibly bind, but it's going to change the shape and speed it up. So if we look at this example here, this is one with just one subunit, but it doesn't matter. It's the same result. So when we look at this allosteric inhibition, you can see that when we bind to the enzyme somewhere here, we're going to change the shape of the active site, and now this substrate molecule can't bind, so we're going to kind of slow down or stop the rate of the reaction. Whereas with this allosteric activation here now, what we've got is binding of this activator is going to change the active site in a way that's going to kind of speed up these chemical reactions. So you can see we have inhibition, we have activation, and allosteric, again, is just the change. We're changing the shape of the enzyme. All right? So now moving on to feedback inhibition. With feedback inhibition, what we've got is we've got some sort of a metabolic pathway that's being controlled by the end product of that pathway. So as the end product kind of builds up, we're having a higher concentration of it, we don't have any reason to make anymore. So we're going to either slow or stop that particular pathway. Okay, and there's a couple of different ways that it can occur. It can occur kind of in the pathway itself involving the enzymes and the metabolism, or we can inhibit and kind of change the activity level of a particular gene. Okay, and basically what's happening, not always, but oftentimes, and this is the example we're going to use, is with this feedback inhibition, we're going to switch off that pathway when that end product builds up. The accumulation of the end product is going to slow or stop an enzyme early in that pathway, so we don't run the pathway all the way down to, say, the second to last step and then shut it off, because we'd be wasting a whole bunch of resources kind of in between. So generally, again, that end product is going to stop the enzyme very early in this pathway and shutting it down. A commonly used example here is with threonine, and what we're going to do is we're going to use some threonine and we're going to make isoleucine out of this. So these are two amino acids. And what happens is the threonine binds and we start to increase the amount of isoleucine here as an end product. And what that does is it's going to allosterically bind to and change the shape of this particular enzyme so that the active site is no longer available. And so when the isoleucine starts to get used up by the organism, the levels drop, and then this thing is going to kind of detach and drop off. And then what you're going to have is that active site's going to take on its native conformation or its original shape and allow that threonine to bind and allow this pathway to run. And this is a very good example of negative feedback, because what we're going to get is, as you see the levels rising and falling, we've got this set point here. And as these levels rise, we get more and more isoleucine. We're going to shut the pathway down and do so by allosteric inhibition or feedback inhibition, allosterically binding to kind of change the active site and slow the process down. And then once the isoleucine starts to drop and get used up by the organism, now we're at a low point, and now we've got a lot of enzymes with active sites that are available, so we can kind of turn this process back on again. And what it's going to do is going to get us back up to the top, where it's going to slow down and stop the process again. So again, this is a good example of negative feedback. Okay, so we've got negative feedback there, and we'll talk about positive feedback, in a different example later on in the course. But this is a good example of negative feedback, just one that you can kind of keep in mind when you're asked about it. Okay, another way that we can regulate kind of metabolic pathways is just use this idea of feedback inhibition at the level of the gene. We can control the gene expression. And one of the commonly used examples involves bacterial cells with tryptophan. Tryptophan, again, is an amino acid, and this is needed by the cells. But when it's needed and isn't present in the environment, the cells can kind of turn on a series of genes and make it, okay, out of some of the stuff that is present in the gene. And when the amount of tryptophan in the cell it increases, when we don't need it anymore, now we can work to kind of shut this pathway down. So you can see, and we'll learn more about like how these pathways work and the DNA, transcription, translation, things like that later on in the course. But just for now, it's easy to just kind of look at and see that we have an enzyme here, RNA polymerase. And what RNA polymerase is going to be able to do is read this gene, make the messenger RNA, so it's going to transcribe the gene, then it's going to get translated into these peptides that are going to be used to kind of manufacture this tryptophan, okay? And if you look over here, you can see that we have a repressor protein that we're going to be able to kind of slip into this operator site right here and control access to these genes from this RNA polymerase. So we can kind of regulate it at this operator site right here, okay? So when tryptophan is in excess, we have lots and lots of tryptophan. It's going to act as kind of a co-repressor. It's going to change the shape of the repressor protein, and it's going to prevent us from being able to manufacture tryptophan because we don't need it. So you can see here, tryptophan, when we get high levels of it and it binds to this regulatory protein, we can see here when it's inactive, you can kind of see this in this hypothetical little model right here. It's not in a shape that's going to be able to bind, but as soon as tryptophan is present, what we're going to be able to do is slide it into there, and it's going to block access to these particular genes over here, and we're not going to manufacture any of the tryptophan because it's already present in high amounts. And then when it starts to get used up, then it's going to drop off, and then we're going to be able to manufacture more of it. Okay, so as these tryptophan levels fall, this repressor protein is going to take a form now that can no longer bind to, so we're going to kind of work it in reverse. As the levels begin to drop, we kind of move into this shape. Now this cannot bind to the DNA, and then now we're going to be able to make more tryptophan. So again, that's a way that the bacterium in this case is going to be able to regulate the amount of things that it has present in its environment. All right. So another form of feedback inhibition that bacteria use involves lactose. So it works a little bit differently than that tryptophan regulation that we were just talking about. So in a bacterial cell, lactose is a sugar that's consists of glucose and galactose. It's a disaccharide. You probably remember when we went over that earlier in the class. But when lactose is absent, when we don't have any lactose, we don't have any need to make lactase. We don't need to waste the energy to make an enzyme for something that's not available. Okay, so what happens is you can see this repressor protein is bound, it's locked in place, and it prevents us from being able to make any of the lactase protein. Okay, but when lactose is available now, we also have a substance called allolactose that is also available, and this is going to kind of act as an inducer. So what this is going to do is this is going to be able to bind, this allolactose is going to be able to bind to this red repressor protein here, and when it binds to it, it's going to change its shape, and what it's going to do is it's going to cause this. Now you can see when it's binding, it's making it inactive. So what that allows now is this machinery to kind of read this gene and make the substances down here that are necessary in order to break down lactose. So basically what this is lactase, what we're able to make with these purple things here, and there's multiple things involved, but we're basically making the enzyme that's able to break down lactose. Okay. However, in this particular case, it's kind of only part of the story. So what's going to happen is if we have a case where we have both glucose and lactose available, glucose is more preferentially used because it's not a disaccharide. It's more easily used by the organism, so it's not going to expend any energy to make lactase because it's got some glucose available. And that's going to be preferentially used, and we don't need to activate any of those genes. So you can see here, when lactose is present, Okay, the repressor is, inactive, so we could conceivably make the genes. However, glucose is also present, so we don't have any reason to express these genes. We're not going to waste any of the energy. We've got a preferential form of energy available to us in the glucose. Okay, so what's basically going on is when we have glucose, we have this regulatory molecule called cyclic AMP or CAMP. Okay, and CAMP is necessary to activate a regulatory protein to turn on the lactase genes. So in other words, the CAMP levels and the glucose levels are inversely proportional to one another. When the glucose levels are high, CAMP levels are low, and we don't express any of the lact genes. However, when the glucose levels are low, now the CAMP levels are high, and then what we're going to be able to do is activate these genes necessary for breaking down the lactose. Okay, so when we look at this now, this is a case where we have lactose present, but glucose is scarce, and because we don't have much glucose, we've got a lot of cyclic AMP that can bind to this activator protein, bind to the DNA and activate this RNA polymerase so that we can express these genes. Okay, so this is a case where lactose is present but glucose is scarce. Now we're gonna, we want to activate those lact genes so that we can make the lactase and break down the lactose and get the energy out of it. All right, so regardless of the pathways that we're talking about, both of these pathways are ways that we can regulate the metabolism. We can do so, as we saw right here, with this first one, by regulating the enzyme in the pathway, changing its shape and preventing it from working as the tryptophan builds up. But when the tryptophan kind of concentration goes down, then now we're going to be able to drop off that enzyme. It's not going to be present in the mixture, so to speak, and the tryptophan drops off. The enzyme takes on its native conformation. It allows us to synthesize more tryptophan. Again, that was the example of the negative feedback that we talked about. And the other way that we can regulate the metabolism here is really at the level of the gene. And what we talked about was the lactose, different ways in which we can metabolize the lactose. So what we're basically doing is we're preventing, by having what we need present in the reaction mixture, we're going to either allow for or inhibit access to the gene in the manufacturing of the machinery necessary for kind of taking advantage of the stuff that's in the reaction mixture. Okay, so we basically went through these two things, and in its simplest terms, we're either going to regulate the enzymatic pathway or we're going to regulate the expression of the gene. Okay, so I know there was a lot here. We discussed a lot of this in class. I told you I wanted you guys to watch this video, and then what I want you to do now is, if you have any questions, and I'm sure you probably do, write down the questions or highlight the particular spots or whatever the case may be. Just recognize the slide, and we'll definitely go over these again in class. Okay, so this is something that's kind of worthy of talking about, things that you want to know as far as like how metabolic pathways are regulated and some of the reasons why. Okay, so make sure you write down your questions, and we'll talk to you soon. Take care.